Welcome to the second activity, which is about the chemical transformation of starch. You remember in grade 5 when we asked a question about when you introduce a slice of bread inside your mouth, it mixes with saliva. Then a slight sweet taste is obtained. We are going to perform this experiment outside our body, that is in vitro, of digestion of starch. Digestion in vitro of starch. To 2 test tubes containing starch, I'm going to perform iodine test to indicate the presence of starch. Iodine water is added to the 2 test tubes. You see, the color of iodine is brown orange. It turns dark blue in the presence of starch. The 2 test tubes contain starch, tube A as well as tube B. Now we are going to add fresh saliva to tube A only. Fresh saliva is added to tube A only, and the 2 tubes are placed in the water bath, which was adjusted at 37 degrees Celsius for about 20 to 25 minutes. 20 minutes later, Fehling test is applied to test tube A. You remember the test tube which contains fresh saliva and cooked starch. To this tube we are going to add an equal amount of Fehling solution, which is blue, and heat the mixture. Okay? You see, the blue color start to reduce, indicating that starch is transformed to something else. You remember when I said a slight sweet taste is obtained? This slight sweet taste is a sugar called maltose. So starch is transformed into maltose in the presence of saliva. You see, the color, the blue color is reduced and transformed into any color. Rather than blue, indicate a positive result. Upon applying Fehling test to test tube B, you see, the blue color remained unchanged, indicating that the absence of a reducing sugar. So starch is transformed into maltose in the presence of saliva. In fact, saliva contains a chemical substance which is an enzyme. This enzyme is called salivary amylase, which gradually breaks down starch into maltose sugar. A complex sugar is transformed into a less complex sugar, which is maltose, by an enzyme called salivary amylase.